Hello, what's good everybody, and welcome back to the channel. If you're new here, you just tapped into Ryan's science lessons, and trust me, you're not going to want to miss this. Today we're diving into the phospholipid structure. This is Mr. Razai back with another banger. Today I will be breaking down the phospholipid to you. In orange is the head of the phospholipid. The two squiggly lines are the tails of the phospholipid. The head is polar, which makes the head hydrophilic. Hydrophilic basically translates to loves water. The tails, however, are opposite to the head. The tails are non-polar, which makes the tails opposite of hydrophilic, which is hydrophobic. And hydrophobic basically translates to hates water. It's like one end of the phospholipid loves water and the other end hates water.
Kind of like me at a family reunion. Ha 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 ha.
Now moving on. The head makes up the phosphate region, while the tails make up the fatty acid region. Now, when making the phospholipid bilayer, the head of the phospholipid basically faces the aqueous environment or the water-filled environment, while the tails face inwards so they don't interact with the water. And guys, that's a wrap. Thank you for watching. If you enjoyed this show, please smash that like button, hit subscribe, and ring the notification bell so you never miss an adventure with us. Drop your thoughts in the comments. I love hearing from you guys. Thank you and stay curious.
My name is Michael Chang, and I'll be talking about the plasma membrane structure and its components. This is a simple diagram of the plasma membrane structure, and I'll be going over the phospholipid first. One of the main structures of the plasma membrane is the phospholipid. Every phospholipid has, as pictured, one water-loving head and two water-fearing tails. The plasma membrane's main structure is built as a bilayer, which means that it is two layers of phospholipids facing back to back. This bilayer comes from the water-loving heads facing outwards and the water-fearing tails facing inwards. This arrangement allows for a flexible yet strong layer of protection. Next, we will be going over cholesterol and its effect on the plasma membrane structure. This is an example of what happens to phospholipids when there is no cholesterol present and the temperature changes to too warm. As seen in the video, without cholesterol present, the phospholipids become too floppy and lose much structure. This is because the warmer it gets, the more flexible the phospholipid bilayer gets. This is an example of what happens when there is no cholesterol present and the phospholipid cools down. As shown in the video, what happens when the phospholipid is cooled down too much is that it becomes rigid and too inflexible. This diagram shows what the cell membrane looks like with cholesterol. Still sturdy, however not too rigid and still flexible. Next, we will talk about different proteins and their functions. As labeled in the diagram, you can see an integral protein and a peripheral protein. These two proteins have very different functions. Integral proteins, as seen, span through the entire cell membrane and act as hallways for material to pass through, while peripheral proteins offer support and structure to the membrane. As pictured in the diagram, there is an integral protein acting as a hallway to let material through the membrane. There is another type of channel known as an ion channel that reacts to different types of charges opening and closing to allow the passage of ions. Another type of protein is known as a transport protein. These types of protein use ATP as an energy source in order to move material across the membrane. What is being shown in the diagram is known as a sodium-potassium pump, a type of transport protein that moves sodium-potassium across the membrane. Another type of protein is known as a channel protein. These proteins provide passageway for materials that are hydrophilic and polar. Finally, the last type of protein is known as a carrier protein. These types of protein bind to specific molecules and change their shape before moving them through the membrane. And this will conclude my video. On behalf of my group members, we want to thank you so much for watching and hope that you learned a lot.
Cell transport by Mir-Lemons. The first concept we're going to be talking about in this video is, as you see right now, concentration gradient. Concentration gradient is very important in biology and will come up multiple times in the course. So to understand biology and why things happen, it is important to understand concentration gradient, and not to mention concentration gradient is a key concept in this video alone. So it's very crucial to understand what it is. So concentration gradient is the difference in concentration of a substance between two areas. So if we look at here, we have a solution, we have two solutions. So we have the inside of the cell solution, and we have the outside of the cell solution. So as we see in the inside of the cell, we have a higher amount of solutes than on the outside of the cell, therefore creating concentration gradient because there is the difference of concentration. So why this is important is because concentration gradient will determine where the solutes will flow. In a concentration gradient, the solutes will always want to flow to an area of lower amount or lower concentration. So if we see here, we see that, the inside of the cell has way more solutes than the outside of the cell. So what's going to happen is the 33 solutes, not all of them, but some of them are going to flow outside of the cell to try to balance the amount of solutes on the inside of the cell and the outside of the cell, trying to create the even amount of concentration on both sides. So that's why it's so important. The next thing I'll be talking about in this video is selective permeability. Now, selective permeability is just when a cellular membrane allows some things to enter and doesn't allow some things to enter. So here we have a cellular membrane. Cellular membranes are very prevalent in human life and in biology and any living thing. And here we have some common solutes that we may see in life and biology. So to understand this, we have to understand which ones can enter and which ones cannot enter. Okay, so the ones that will be able to enter are going to be H2O and O2. This is because O2 is a hydrophobic molecule, and this is important because the tails of the phospholipids are hydrophobic, making the agreement between them and allowing them to pass right through. H2O is a polar, small, uncharged molecule. Now this is important because the heads of the phospholipids are polar, allowing it to agree and pass right through. Now the tricky parts and the tricky things are the ones that are not going to be able to go through, which would be glucose and potassium. Glucose cannot go through because it's too large to flow right through these phospholipids. It's too large and will get rejected. Potassium, on the other hand, is an ion. This is special because it does not agree with the inside tails of the phospholipids, which are hydrophobic, and they do not agree, therefore not allowing to pass through without a facilitated protein. The first concept we're going to be talking about today is passive transport. Passive transport is just transport that doesn't require any energy. So the first type of passive transport we're going to be talking about is just diffusion. So let's have something that's going to be traveling across the cellular membrane. So here we go. This is something that's just going to be traveling across the cellular membrane. Diffusion would pretty much be this. Let's say this is H2O traveling across the cellular membrane just freely, pretty much, with no energy is required. It's just flowing from one side to the other. We see this when there's a concentration gradient. So let's say there is more solutes on this side than on this side, then this solute's going to flow from that side. So that's where we see it, really. This is where we see diffusion. Now, the other one I'm going to be talking about is facilitated diffusion. Facilitated diffusion is a lot similar to diffusion, but the main difference is that you're going to need a protein in facilitated diffusion. So as you see here, the purple looks like tube here is going to be the protein that's going to be the facilitator for the traveling / transport of this solute. So here you go. We just did some facilitated diffusion right there. What we're going to be talking about is active transport. Active transport is different from passive transport because energy is required for the transport of whatever is being transported across the cellular membrane. So here we have energy. This lightning bolt with the energy above it's going to be representing the energy that's needed for the transport. And here we have a protein, an integral protein. So a good example of active transport would be a sodium-potassium pump. So in this one, we're going to just use potassium as the example. So here we have energy. Let's say this energy is being applied to this protein, therefore making it active transport, and then this potassium is going to flow through here to the other side. Now, where we see this is when something is trying to travel against the concentration gradient. So let's grab a lot of potassium here. Let's bring this over here. So here, we have a concentration gradient. We have 1 potassium versus 5 potassium. So what would happen is, let's say we're having active transport, what would happen is this potassium would actually flow against the concentration gradient to the other side. This is where we would see this. Bulk transport is the last and final thing we're going to talk about in this video. Bulk transport is when a cell is trying to gain or excrete a lot of material. This type of transport does require energy. And the 2 types of bulk transport would be endocytosis and exocytosis. Exocytosis is when the cell is trying to excrete material, whereas endocytosis is when the cell is trying to gain material. The first example we're going to see today is exocytosis. So what's going to happen in exocytosis, we see an internal vesicle that's going to come up all the way to the cellular membrane, and it's going to attach to the cellular membrane and open up. So to show this, I'm going to erase a little bit. This opening is now going to allow whatever materials inside the vesicle to escape and go outside the cell. The next example we're going to talk about is endocytosis, where the cell is trying to gain the material on the outside. So here we see an external vesicle. What's going to happen is it's going to come up to the cellular membrane, it's going to attach, then it's going to conjoin with the cellular membrane, and everything inside is now going to go inside the cell. So I just want to say thank you so much for your time, and thank you for watching. Thank you.